Oh yeah, okay, good. There's a thumbs up. I saw that. So good. Everyone can be on Teams. Good. All right. Okay, we're ready to go. Okay. So um, I'm just finishing up the first part of the lecture on the basics of fMRI, and this is basically, I mean, this is sort of near and dear to my heart, and it's sort of dealing with all the aspects of the hemodynamic response, the dynamics, the post-undershoot, the upper temporal resolution, how to interpret it properly. So um, just going to, oops, okay, that's that. All right, just going forward here. So as I talked about uh, last week, there's many different ways you can detect brain activation with MRI. And the more established ones are flow, oxygenation, blood oxygenation, and blood volume. And blood volume can be detected invasively or non-invasively. And there's a whole slew of other ones. There's spectroscopy, there's other experimental techniques. That's one thing that's really cool about MRI is that uh, there's so many parameters that you can manipulate that we're still in the process of trying to figure out what we can be sensitive to with MRI. And even with bold contrast, and as I'll suggest when I show the dynamics, there's a lot of aspects of the bold signal change that might be sensitive to things that we haven't yet realized. So that might be in there, like excitation inhibition ratios and things like that. So that we haven't yet figured out, and we'll need non-invasive alternative measures to figure that out, or invasive ones. So the previous lecture, I talked about physiology that accompanies brain activation, basics of bold, vein effects, great echo versus spin echo, and also effects of respiration and blood pressure. And in this lecture, I'll talk a little bit about the dynamics, the nonlinearities, the latencies, a little bit about resting state. There'll be a lot more later on, perfusion imaging, CMO2 imaging, and blood volume imaging. So the first thing is that, so this is actually taken out of my thesis, my PhD thesis that I wrote back in 1994, and it was a book chapter that it ended up being. But the first thing to do when you have a signal of any type, what engineers usually do is they probe it with either an oscillation or an impulse or everything in between. And I decided, okay, let's see how rapidly we can turn on and off the brain and still have the hemodynamic response keep up. So back then we just had very basic, I just had motor cortex. And you can see that as you go faster, so the signal starts to represent a sine wave at about 6 seconds on, 6 seconds off. And then finally, at about a duty cycle of about 15 or so, 16 seconds, it starts to dip rapidly. So that's basically a low pass filter. And it no longer, it sort of stays saturated in the on state at about 2 seconds on, 2 seconds off. So this was picked up again, and this is the beauty of fMRI as well, is that, I did that at 1.5 Tesla with motor cortex and, some regular quadrature RF coil. And, people picked it up again. So Laura Lewis and John Polomini decided to probe this a little bit more. And it was very recent, and they were actually able to go faster. So it turns out that they used a visual simulation, and they were able to have a duty cycle of about 0.5 hertz, or 2 seconds on and 2 seconds off, and still detect a deflection in the signal. It certainly became saturated in the on state, but they were able to detect something. And this was surprising because if you assume that the hemodynamic response is a linear system, and it reflects what the input is, and you know the characteristics by the impulse response function, there is no way that 0.5 hertz signal should have been detected in any sort of way. But they detected it. And that's why this paper was so impactful, because it implied that the hemodynamic response is actually very nonlinear in certain regards. And so this is the linearity, and this is where they detected, and this is a linear system right here. So this implies that if you look at these typical hemodynamic response curves, this implies that the hemodynamic response curve, when you're going this fast, is something displayed by this, like this dotted line right here, which is very different than this hemodynamic response curve. So that's still kind of a mystery as to why that is and exactly what that means. But the fact is, it's almost to the point where you can detect neural oscillations, but didn't quite get there. So it's still limited in the use in terms of how fast you go. Okay, and we also probed the effects of stimulus duration. So take one extreme, I would put people in the scanner, and this was and shine flashing checkerboard light in their eyes for 25 minutes straight, and both flow and bold stayed elevated the entire time. So that was actually an unknown in the early days of fMRI. Does the bold signal stay up? One group saw that it went down, and that was sort of a, that caused some controversy, but it turns out the stimulus just basically wasn't compelling enough to keep the signal up. This was just basic low-level visual stimulation. V1 doesn't really care, it just stays working. So that was good. So the very first impulse response event-related fMRI study was back in 1992. And that was by Andrew Blamayer in Yale. And I always like to mention this because he always gets overlooked for first doing event-related fMRI because they didn't call it anything. It was basically 3 seconds on and nothing, and they were able to elicit a response pretty nicely. And then we decided to go faster. This is also work where I worked with the motor cortex. We were able to elicit a really nice response down to about half a second, or I'm sorry, half a second. And you still elicit a response, and it seems like it behaves somewhat as a linear system with a post-undershoot. And going even faster, down to about 250 milliseconds. It seems to go down, but then stay at a certain level at about 250 milliseconds. This is visual stimulation. And even going down, this is some work with Bob Savoy at MGH back in 1995. We even were able to go down to 34 millisecond stimulus, and still we're able to elicit a response. And so the conclusion there is that there is no lower limit. It's a lower perceptual limit of what you could actually activate. So there's no limit to how brief of a stimulus you can give. There is a limit to how small the signal gets. It seems like it saturates in some low level of activation at about maybe a half a second or so. So, and as a side note, there's work with Randy Buckner. He was the first person to do event-related fMRI with more of a cognitive task. This is a word stem completion study in which subjects were seeing word stems and they had to complete it. This generated some excitement that he was able to generate an event-related response in the left anterior prefrontal cortex. And there are other probes as well. This is work by Rasmus Bern, in which he actually tried to characterize the linearity. So if you gave, once again, a linear system predicts that the red line. And so with very brief stimuli, the hemodynamic response is much larger than expected from a linear system. And this was potentially modeled. So this is more quantitative. This is actually looking at the linearity, a measure of linearity as a function of stimulus duration. So only after about like 2 seconds does the hemodynamic response become linear, like behave like a linear system. But what's happening at the very beginning? So we tried to, and also this is also an interesting thing that's still not quite fully understood. And that is if you have the stimulus on the entire time and intermix brief off periods of different durations, the hemodynamic response behaves in a sublinear way. It actually is a superlinear system when you're trying to turn it off briefly. So that, to the best of my knowledge, these mechanisms have not really fully been investigated, even though, it's been about 20 years, 21 years since, I'm sorry, wow, 21 years. since that was looked at carefully. Wow. Um, One explanation for the nonlinearity, though, with very brief stimuli, is that at the very beginning, if you have an abrupt onset with neural activation, at the very beginning of that onset, there's usually a burst of spiking. So one way of thinking about it, this is some seminal work by Logothetis, and he showed that that the hemodynamic response behaves very linearly, once again, with a contrast, with a checkerboard that varied in contrast. So the more contrasty it became, the hemodynamic response went up, and the electrical activity as that was simultaneously measured, or the local field potentials, went up. However, what he didn't really mention is that, to connect this point with zero, which you have to do, the system is incredibly nonlinear to go from here to here. And the reason for that is because of this big spike at the very beginning, potentially, that shows this sort of electrical response that needs to, that's involved with the hemodynamic response potentially. So we tried to model this. Rasmus Byrne put this into a simple model of having, let's say, a brief burst of activity at the very beginning, and we got pretty close to modeling what was expected from the hemodynamic response, although you really had to have a little bit more neural activity than expected at the very beginning to really fully characterize the nonlinearity, this nonlinearity in the first 2 seconds. One thing we also noticed, and this actually has been written about, I don't have the reference right here, by Kellenic Grill-Spector. She's done some studies on this as well, that near the periphery, so it seems like this nonlinearity has a spatial dependence as well. So near the periphery of the visual cortex, you have a little bit more nonlinearity. And once again, this is probably reflecting neuronal activity. There's nothing potentially nonlinear about the hemodynamic response. Oh, yeah, yeah. Oh, well, I mean, a linear response. So backing up a little bit. And so if you convolve a hemodynamic response with a boxcar function to get your activation, a linear response is just basically you're allowed to do convolution at all different durations, and it would behave a certain way. It's independent of the duration of the stimuli. This is one way of looking at it. And so with a linear system, you have this red line right here, that if you have a very brief response, you convolve that with a hemodynamic response, it will show this small activation. And then it grows as the duration goes up, which is expected from a linear system. With a nonlinear system, it might show, and this is the case, a much larger than expected response with a very brief stimulus, meaning that there's some amplified transfer function at very brief stimuli. But that seems to not apply as the stimuli get longer and longer. So that there's something highly nonlinear going on. The system is nonlinear for very brief stimuli. It also turns out the system is nonlinear for very weak stimuli. And I don't have this data, but there was some work that showed, yeah, I don't have it, but basically showing that with simultaneous EEG or electrodes with attention, they found that for very low levels of attention, it turns out that the bold response sort of overrepresented the level of activity that was sort of represented by low level activity with attention studies. So basically it was anything that's a weak stimulus seems like it's overrepresented, either brief or very weak. Bold slightly overrepresents that. So, and here's another example of nonlinearity. This is a study we did a long time ago with post-bac in our lab, August Tuan. And this was back in 2008, where we did MEG studies along with fMRI studies and showed, this is the MEG plots and this is the fMRI plots. If you abruptly turn a stimulus on, you always get this transient. And with MEG, you notice this as well. And this was sort of a flashing checkerboard of about 10 hertz. Yeah, 10 hertz. But as you increase, if you decrease that from an abrupt onset to a more shallow ramp all the way down to a very shallow ramp, you don't get this abrupt effect in the bold response that contributes potentially to the nonlinearity at the very brief stimuli. And you can see that reflected also in MEG as well. So that was a nice sort of comparison. However, because the system does behave linearly from 2 seconds onward, or maybe a little over a second onward, you can do a useful mathematical technique of basically deconvolution to get the neuronal responses. So you can, so if the stimuli, this is a nice work by Anders Dale, way back in 1994, 5, I believe. So basically showing a flashing checkerboard and then showing 2 flashing checkerboards, spaced about a second apart, and then 3. And the question was, from this, composite response with the 3 flashing checkerboards, can you subtract out the combination of the 2 or the 1 to get back your original hemodynamic response? And that's another test for linearity in some sense. And they were able to get it back just fine. So because of that, that evolved a paradigm that was wonderful. That was basically that we use all day, all the time, even today. And that is randomized event-related. So jittered event-related responses where you, instead of giving a block design where you give, responses for 30 seconds or so, then off, then on, and then instead of giving like an event-related where you have to wait another 15 or 20 seconds until the hemodynamic response completely dies down, you can go very fast. You can go very fast as long as you jitter the response, you randomize the response, and what you can do then is deconvolve the stimuli according to each of your conditions from that randomized response. And this was an example where he showed basically a constant ISI. If you're constant at 1 second, it just is, there's no variation. But if you jitter your if you have an average ISI of 1 second interstimulus interval, you get a lot of variation that you can then deconvolve and get this hemodynamic response back. So that was sort of a major advance in fMRI. And then the question was, what's the optimal speed to go? And there were several papers published in parallel along this line. Anders Dale published one. Tom Liu published one from San Diego. We published one. Rasmus Byrne published one, which I'm biased, but I like this one the best because it gave 2 conditions. One, if you're just trying to map the activation, you want maximal sensitivity. And of course, maximal sensitivity usually is attained with a block design. And so that's considered the best. But if you go shorter and shorter in your stimulus duration, your optimal interstimulus interval is about double the stimulus duration. But it's randomized. So this is randomized and then deconvolved. And so you can see how they compare. So the longer you go, the more contrast you get. But for a given interstimulus interval or stimulus duration, you optimize. For instance, stimulus duration of 2 seconds, the optimal ISI is 4 seconds. And it's assumed that you're randomizing that input. However, and this is what most of the field kind of misses, and it's always interesting. Let's say you know where you're looking, and you just want to characterize the details of the hemodynamic response. And so you take a region of interest, and you're just looking at that hemodynamic response, and you just want to understand that one hemodynamic response. Then it's better to go faster. So it's better to have a shorter stimulus duration and a shorter average ISI. And the faster you go and the shorter you go, the better off you are for characterizing that hemodynamic response once you know where to look. But if you just want to detect, if you just want to make a nice map, then it's better to go slower. But if you need to go fast, this is the relationship. So that's pretty much all I wanted to say about that. All right, there's other aspects of the hemodynamic response. There's the pre-undershoot and the post-undershoot. And, this has caused the field all kinds of discussion. Pre-undershoot was suggested to show an increase in metabolic rate before the flow had a chance to kick in. And the post-undershoot, which is much more robust. So the pre-undershoot kind of died because nobody could really see it. Nobody could really characterize it. So we gave up on that. It still shows up every once in a while. However, the post-undershoot is robust. And people have tried to model it mathematically with a double gamma variant or with another one. But they've also, more importantly, tried to model it using physiologic models. And it turns out that a physiologic model that includes, this was the balloon model by Richard Buxton, and it claims that if you have for instance, a perseveration of increased metabolic rate, you can produce a post-undershoot because you have more, after the flow has gone down, you extract more oxygen from the blood. If you have a perseveration of the blood volume, you can also produce a post-undershoot. And if you also have an undershoot in the flow, you can produce a post-undershoot. So there's 3 different ways of producing a post-undershoot. And so then that became like, grist for the field to discuss and debate for a long time. And it turns out that it's probably, the post-undershoot is probably a combination of either blood volume or longer metabolic rate. But it's not fully settled. Okay, and there's other mysteries too. Like, for instance, the mystery I told you with the extremely rapid duty cycle, we're able to detect something. And so the whole hemodynamic response has to be different. And why is that? We don't know. Turns out, as expected, potentially there's differences in how much the signal habituates if you have a constant visual stimulation versus checkerboard, which is expected. But there's a bigger difference in the post-undershoot that occurs. Also, a curious thing is that with and I talked a little bit about this last time, but if you add diffusion weighting to get rid of the intravascular signal, the blood signal in the vessels, about a third of the signal disappears because that's from the inside the vessels. But the post-undershoot isn't affected at all, which implies that that the intravascular signal doesn't contribute to the post-undershoot, which seems like it implies that the oxygenation is back to normal again, and so it's a blood volume effect or something like that. So that's another mystery. And there's also a lot of information that we're still gathering that we still don't fully understand. And I have to emphasize this plot was made like now almost 30 years ago. And I looking at the literature, and maybe of course I've probably missed something, but there's a lot of systematic differences in the post-undershoot. And this is just one example. So if you have a flashing diffuse red stimulus, and then you alternate that with a black and white pinwheel, for instance, you might get the same level of activation, but huge differences in the post-undershoot. So there could be ongoing, inhibitory signal or differences in the neurovascular effects that occur in the post-undershoot with the stimuli. So there's all these transients. And I guess the message I want to convey is that there's many different transients in the signal. There's the onset transient that I talked about. There's the post-undershoot, and everything in between that contains information. And this information is still being plumbed for useful insights into neural activity or hemodynamics. And not many people are working at it. You have the cognitive neuroscientists doing their thing, and then you have the physicists doing their thing, and somewhere in between, nobody's actually probing this that much. So it's a huge opportunity. Okay, let's talk a little bit about latencies. So, one of the goals, one of the things that people in fMRI became excited about potentially was like, oh, well, if the hemodynamic response goes up and if it's well behaved everywhere in the brain, then you might resolve one area versus the other area, show it turning on. And people try to do this all the time with things like, Granger causality analysis, or just looking at the latency, saying, oh, well, if this turns on first, then this happens. Problem is, and this is a very early study, once again, from about, almost, 28 years ago. Wow, it really makes me feel old looking at these now. So anyway, but 28 years ago, this was a basic study of bilateral finger tapping, just tapping your fingers, left and right fingers. And you'd expect that the hemodynamic response would behave, it all goes up, left and right motor cortex, just fine. But a very simple way of probing latencies is just taking your canonical hemodynamic response. This was event related, so we tapped for 1 second, and just shifting that forward and backwards in time and finding where the correlation is highest on a voxel-wise basis. Very straightforward. It's been around forever. And what we find is that the number of voxels that show, if you plot the number of voxels versus delay, you see a plus or minus 2 seconds in the hemodynamic response. So that's 4 seconds of spread in onset time in the hemodynamic response due to the plumbing in the brain, not due to neuronal activity, due to just purely plumbing. So the idea is that, with activation, flow goes up, you have activation of the capillaries, then it goes downstream into larger vessels. So the larger vessels typically have the later responses. And so this variation is 4 seconds. You can't really do much to resolve neural activity with a 4-second spread in latency. People have tried to resolve this. One thing you can do is that because the hemodynamic response is so well behaved on a voxel-wise basis, even though there's a spread in the hemodynamic responses, it's really well behaved. And one simple experiment that I did a long time ago was with Bob Savoy at MGH was simply give us alternating checkerboard, turn on one hemifield and then turn on the next, and delay this by 500 milliseconds. But the key thing is that when you turn it off, you also delay by 500 milliseconds, and then do it again and then delay one side, the other side by 500 milliseconds and turn it on. So you're looking at the stimulus timing relative to itself with a different paradigm, with the modulation of the paradigm timing. And what we were able to find is that we look at the two regions, we're able to find a little bit of a difference in the onset. The signal is much better behaved when you're driving it than when you're not driving it. So when it turns off, it gets more noisy as it goes back down to baseline. When you're driving it, it's well behaved. But this didn't really translate to the map. So if you have a map of 500 milliseconds delay between left and right hemifield, you can't really see much at all. You're dominated by the variation in the hemodynamic response, plus or minus here 2.5 seconds. But if you switch the order, you still can't really see much. But if you subtract these two maps, it comes out pretty beautifully that you can actually see the onset difference. In this case, it's a second onset difference, 500 and 500. And you can go faster. This is 250 milliseconds. You still see a difference. And Ravi Menon a long time ago with his paper on mental chronometry, published it in PNAS, he went down to 100 milliseconds. So the message here is if you modulate a task, if you look at a task and then modulate the timing a little bit, you can discern a modulation of down to 100 milliseconds. And it really is very much a function of not the hemodynamic, that doesn't have anything to do with the hemodynamic response being slow. It all has to do with the hemodynamic response being, having a little bit of noise in the signal. If your signal noise was infinite, you would be able to resolve to infinite resolution temporally if you modulated the stimuli. It's all a factor of the noise. So when people say, oh, well, because the hemodynamic is slow, we can't resolve things. No, that's not the right way to say it. It's because the hemodynamic response varies over space that you can't resolve things absolutely. And it's because it's noisy that you can't resolve lower than 100 milliseconds in the onset timing if you modulate the stimulus. And so, but this can be applied really nicely to cognitive tasks. So this is work by Pat Belgowan when he was in my group, in which you had a subject view either nonsense words or rotated words. And it turns out that if you looked at words, if you compared the timing between words and non-words, there was no difference in the onset in the left anterior prefrontal cortex for processing words. But for the non-words, it took a little bit longer. And so the hemodynamic response was a little bit wider. Turns out, though, that if you looked at the delay that occurred naturally with rotation as a function of how much the word was rotated, and it turns out you can't really see it that well here, but that with rotation, the onset of the left anterior prefrontal cortex was delayed because it was this area, then that infers that this area is downstream in terms of processing. So there's another part of the brain that's doing the rotation, feeding it to the left anterior prefrontal cortex that then determines whether it's a word or non-word. So that's the onset's delayed. So you can make some inferences as opposed to where it is in the processing stream based on the delay. Lastly, another way of probing the timing is really nice work that I actually think also should be picked up upon more. This is great work by Seiji Ogawa back in 2000, in which he was looking at left and right forepaw of a mouse. And if you stimulate one forepaw, typically there's a signal that goes across and inhibits the other forepaw. So it turns out that if you do that with a with a long delay, then the effect isn't there. If you do it with too short of a delay, the effect isn't there. But if you time the delays such that it's right around 40 milliseconds separate, that's when the optimal inhibition occurs. And so you can actually measure that 40 millisecond time that occurs, that it takes from left to right motor cortex or sensory cortex to inhibit that other signal. So you have these two slow signals, and all you're doing is looking at the amplitude as it's modulated by the timing between the left and the right. And you get this dip. And so you're able to probe, 50 millisecond delays. And you can even go faster. So this sort of summarizes the temporal resolutions. I'm not going to go into any detail. With techniques such as MREG, now we can scan up to 100 images a second. It's usually not worthwhile at all to do that because you give up signal to noise a little bit, but sometimes it's good for probing details. And it turns out that the fastest you can go, I said 1 second on, 1 second off, probably that's a little bit faster than you can go. You can probably go about, I think that the NGH group went up to 0.75 hertz and still elicited a response. So, all right, let's briefly talk about the noise. And this will be very brief. So, we all want signal to noise when we're imaging. And the thought is, let's give better RF coils, let's go to higher field strength. But it turns out, if you put a phantom, and what I mean by a phantom is like a bottle of water. If you put a bottle of water in a scanner and you image the signal to noise of those images versus the temporal signal to noise, you'll get a nice straight line, a nice line of unity. So meaning that if the image has a high signal to noise measured between looking at the signal in the phantom versus outside the phantom, that will correspond exactly to the signal as you measure over time, the variation of the signal over time. So, but it turns out it's very different when you put people or anything living in the scanner. If you do that, the signal to noise could be very high for your RF coil, but the temporal signal noise doesn't really get up that high. At the most, it's about 100 to 1. Here, actually, this was about 200, but this is an early study. So basically, physiologic noise dominates. And Jerzy Perduka in our group actually put more of a quantification on this. And actually, this was done also even earlier by a person named Gunnar Kruger, who won the Young Investigator Award for this sort of study. But so basically showing that in white matter, gray matter, CSF, there's a upper limit to the temporal signal to noise that you can get. And in gray matter, it's about in the range of 100 to 1. That's it. And that's because of physiologic noise. And this plot summarizes this right here. So thermal noise, if you increase your RF coil quality or the field strength or whatever, thermal noise keeps on going up. Temporal signal noise goes up if you have something that's just a bottle of water. But if you have a living system, you have physiologic noise that puts a cap on that. And so your optimal for fMRI would be to increase your resolution, which lowers your signal to noise, or push things such that you're right at the elbow of this curve. So where thermal noise equals physiologic noise. I mean, certainly for super high resolution stuff, we're usually down here in the thermal noise regime, which is not the best because you have low signal noise, but it's not that bad because we can actually filter it easier using other advanced techniques such as Nordic or whatever. So it's not that bad. But if you are, imaging at super low resolution, for instance, and thinking you're going to gain some signal noise, it's not the case. You're never going to go higher than what the physiologic noise limit is. People have tried, and to this date, people have tried to filter that out, to create filters for the cardiac function or for respiration. And oddly, it doesn't work that well. I mean, there's retro I-core, there's other things. They might push this curve up maybe 10%, but it doesn't get rid of it. So one of the sort of holy grails, that goals of fMRI is to be able to understand our physiologic noise so much that we get rid of all of it, that we can then suddenly have, if we have a signal to noise of 1,000 to 1 with an RF coil, we can have a temporal signal to noise of 1,000 to 1. And we're not even close to there yet. So we're trying to understand the physiologic noise better to filter it out. Okay, but what can we gain from an aspect of this physiologic noise? Well, it turns out there was this breakthrough in fMRI that occurred in 1995 that was kind of ignored by the field. The whole field ignored it for 10 years, and then it finally picked up on it. And that was Brat Biswal, and he was just sort of exploring fMRI and using this technique with AFNI, before it was called AFNI, of just taking a voxel, seeing the time course and seeing how it correlates. Just he was trying to probe the noise, and he found this consistent pattern. And that is, if you took a seed voxel from the left motor cortex, he always found that the area that correlated most with, when the subject was just lying there doing nothing, the area that correlated most with the left motor cortex was always the right motor cortex. That's odd. And so, he would do a task to show that these are the motor cortices, but then this is just random signal this is an example of it, and it always correlated. And so that was kind of a breakthrough. Nobody understood that resting state fluctuations correlated across functionally related parts of the brain. So that was huge. And it's kind of interesting that because everyone was so caught up with doing task fMRI, they just flat out ignored this for 10 years. And also because we didn't really have the tools that were readily available for easily doing this sort of analysis, which wasn't that complicated, but just wasn't there. So since then, there's been nice work. This is Randy Buckner actually it's a nice way of showing as you move around your seed voxel in your brain, you can see how different parts of the brain show correlation that are very much related to how the area is speaking to the other areas. So you move that seed a little bit, just a little bit, you find, all kinds of correlated areas. Well, you can't just look at movies like that. So there's two ways that have sort of dominated for looking at resting state. One is taking a seed like that and then seeing what correlates in the rest of the brain. And that's sort of an hypothesis-driven area. You like take a seed, see what correlates. Although it's extremely inefficient. Another way is to parcel up the brain. And there's various approaches to doing this and making a matrix of every parcel as it correlates with every other parcel. And then making some inference from this matrix. Whether you're doing some sort of population or individual or whatever, how this varies. And then you can also display this matrix. You don't have to do a matrix. You can do these various circle plots. Each area corresponds to a parcel. You can do, balls and sticks that connect it. You can do other things like that. So there's many different ways of displaying that, but those are the two predominant ones. And part of the challenge is coming up with the right parcellation, because it turns out, varies across a lot of people, from individual to individual, and it turns out it might even vary over time. So that's an interesting area of inquiry. All right, so here's some examples. Another way of doing analysis, and this is partially a way that the parcels are generated, but not exactly, is just to independent component analysis. And this is still done. And if you do that, you can find that there's many different networks, independent components that come out from the time series data. And as I mentioned, there's many ways of generating parcels. The ones that are most popular are the Yao Atlas. There's a Glasser Atlas. Matt Glasser actually, by the way, won the Young Investigator Award this year with OHBM, or the Early Career Investigator Award, partially because of this atlas that he created. So there's many different other ones, and there's many different ways of making it, which I'm not going to get into. Okay, so now we have an interesting story here. So now we have neural activity that is due to thermal noise. Time series fluctuations that are due to thermal noise, motion, scanner instability. They're less interesting. And then you have time series variability that's due to breathing changes, which I didn't really get into, and variable end tidal CO2, things like that, cardiac pulsation. Then you have the interesting spontaneous fluctuations that are either at around 0.2 hertz or lower, that are due to underlying neural activity and conscious thought, unconscious processes, neurological vigilance. And the thing is that, and this is sort of an ongoing mystery as well. I mean, fMRI is very practical. We're using resting state fMRI all the time, and we use it for all kinds of things, make all kinds of inferences. We don't have a clue, like, why it's there. We really don't. Why is the brain? Why does it show this spontaneous correlation? But it works, so that's okay. So there's been more papers on explaining why it exists. And I just want to pause for a second. And there's a beautiful paper by Pizzullo. And this basically is saying that the low dimensionality of spontaneous neural patterns may reflect slow transitions between generic priors tuned for low dimensional general purpose behavioral patterns and dynamics across sequences of brain states that may reflect a maximization of entropy of explanations. So basically, in this, or the exploration of the brain's dynamic repertoire that generate models in the absence of data to explain and remain flexible. So basically, the idea there is that during the day, during every moment, you're doing tasks. You're moving your hands in some way. You're processing the world in some specific way, and it's a lot of activity going on. Some of it's coordinated, and then when you're sleeping or resting or doing nothing, those areas are basically settling in as they communicate with each other into these lower dimensional states, and the brain is saying, okay, so let's find these lower energy sort of low dimensional states so we can then anticipate when we have to do this again in the future. That's very hand wavy. And that's about where it's at right now in terms of understanding what they mean. So an example that I always like to show, this is a study where I actually, we did at 7 Tesla, just putting a seed voxel along the motor cortex and going back and forth down and up the motor cortex. And you find, as you move it down, the opposite motor cortex is correlated. However, it turns out that the only area that's the least coordinated seems like the hand area. And everything else seems like it's more correlated. And the idea there is that when you walk, your feet are always moving in synchrony. When you're doing most things, like I'm talking, the left and right side of my face is moving in synchrony. But hands don't really always move in synchrony. They don't always go in a coordinated fashion. And so the thought that's worth exploring is that this dip in the correlation represents the fact that your hands don't always work in synchrony, but they work a little bit in synchrony. So they're somewhat correlated. So the measure of correlation might be a function of how much the brain is coordinated in its activity. Another really landmark study was by Newbold at WashU just recently, about 6 years ago, in which they put a subject and they had volunteers cast their arm, put their arm in a cast, completely immobilizing one arm. And they found, and also the hand as well, they found that within hours, the correlation between the left and right motor cortex dropped. So almost immediately it dropped. So you would think, okay, so you're not using your hand, but still you would think that there would just be ongoing activity. It quickly went down. So was sort of a curious thing that might suggest that what you see at resting state reflects kind of what you've done even hours beforehand. So, which is interesting. All right, that's all I'm going to talk about resting state. In the last 5 minutes or so, I'm going to cover perfusion, which is pretty straightforward. Hardly anyone uses it for fMRI because it's so low signal to noise, but it's still useful. Back when fMRI was discovered, there were a couple of other techniques, mostly initially used for angiography. And the idea there, and I'm totally switching gears now. The idea there was you tagged the inflowing spins, the inflowing blood, with an RF pulse, kind of like giving an injection, but you gave an RF pulse instead. And then you imaged it. Then you imaged this plane as it flowed in. And then as a control, you tagged the opposite side and imaged the plane. And it turns out that if you didn't wait very long after that tag, you only got the most rapidly flowing blood, which was like veins. So you got these big vessels, which was angiography. But it turns out the longer you wait, up to a second or 1,200 milliseconds, you get these beautiful maps of capillary perfusion. And there's different acronyms. This is called flow alternating inversion recovery. This is called echoplanar imaging with signal targeting and alternating RF. Sanji Kim is known for FAIR. Bob Edelman is known for Epistar. Ken Kwong actually also had another technique, and there's more techniques now, quips and so on, that involve various tags and involve quantitation. And this is sort of developed, believe it or not, this was one of the first studies was headed up by Alan Koretsky. He had a very early perfusion imaging technique. As I mentioned, Edelman and Kim and Kwong had their other techniques. These are better maps than what they showed. It's gotten very good at looking at quantitative perfusion. And that was Alan Koretsky and John Detra with their technique, 1992. But it turns out, as I mentioned, the signal to noise is very low. This is actually just a quick comparison of bold versus perfusion. One advantage of perfusion imaging is that you get baseline perfusion, which is great. But it turns out if you take the same amount of time for collecting bold contrast with left and right motor cortex versus perfusion, and basically subtracting these maps of rest versus activation, you can immediately see that the signal to noise is lower. It's lower by about a factor of 4. And however, you can see that with bold activation, these hot spots are right near these big vessels. With perfusion, though, it's very insensitive to large vessels. You are right where there's true perfusion, which is the capillaries, where there's true exchange of spins between the capillaries and the tissue. So that's very different than where these hot spots for large vessels are. So it's lower contrast, but it's more specific. And it turns out, and this is what I inferred my last talk, was that it's also super stable. And this is a plot of sensitivity versus your on-off frequency. And as I mentioned, if you go really fast, things get not very sensitive. But if you go, and for this for bold contrast at its optimal, which is maybe like 20 seconds on, 20 seconds off, the contrast noise is good. But that if you go slower, it goes down because the baseline drifts all over the place. And there's ways of correcting it, but it's never perfect. But this baseline is rock solid with perfusion. So this perfusion is way better than bold if you have super slow stimuli. And this just shows it right here. This is comparing perfusion with bold. And this map is actually most telling, is that even if you compare 24 hours, we collected a baseline perfusion one day, activation perfusion the next day, you get really nice maps of activation. So you couldn't do that with bold at all. All right, the very last part here I want to talk about is CRMO2. So we have, oh wow, 3 minutes left. All right, I'll go very fast. This technique is almost never used, but it's just good to know. If you collect perfusion and bold contrast simultaneously, and there's ways of doing that. It turns out that as you understand with bold contrast, is that with brain activation, the CRO2, the metabolic rate goes up a little bit. It doesn't go up that much, such that the oxygenation overall goes up, but there is some oxygen being pulled from the blood, and that's lowering or blunting the bold signal. So if you give something like a global stress, like have them breathe CO2, the signal will go up, and it won't be blunted by increased metabolic rate because the brain isn't increasing the metabolic rate. So what you can do is compare with comparable perfusion changes or flow changes with a global stress versus activation, and compare the ratio of flow to bold for both. And it turns out with activation, as you can see, the difference between flow and bold is much greater for activation than with with the global stress. Here they're kind of matched, and here there's this big difference, and that difference is due to the increase in metabolic. Map that and create really nice maps of changes in CRMO2 by looking at this calibrated difference. So it's not used that much because it takes a long time, and it's noisy because you're involved in subtraction or division of two maps, but it's still useful if you care about quantitative metabolic rate changes. Last technique, and if you can just wait 5 minutes, I can give you the last technique. It's called vaso. It was a technique that was invented in 2003 by Hanzeng Liu. Oops, I'll get rid of that, close. Oops, there we go. Okay, Hanzeng Liu created this technique. It basically involves where you give an inversion pulse, and 180-degree RF pulse. And when you give that, then once you give that, all the signal is slow, the longitudinal magnetization is slowly recovering. So tissue is characterized by its T1, which is the longitudinal magnetization, and its T2 or T2 star, which is its transverse magnetization. So if you give a 180-degree RF pulse, signal slowly relaxes, and it turns out because of the longitudinal relaxation, and it turns out blood has a different T1 than tissue. And it's different enough such that you can wait until the signal, it goes negative, and then it goes to 0, and then it goes positive. It turns out if you wait until the blood goes through the null point or zero, that tissue has already gone beyond the null point. So there's a little bit of signal in the tissue. Then you give an RF pulse, an image, and then you give them your 90 pulse and you image. And it turns out that if you do that, you get a signal void where there's blood. And if you have an increase in activation, then you have a larger area where there's a signal void because it's an increase in blood volume. An increase in blood volume gives you a larger signal void where there's a null point where there's blood. And it turns out that the signal goes down with vaso because you have this larger null point relative to bold. So that was discovered in 2003 or whatever, or invented. But then it took a while before we realized that it's incredibly useful for ultra high resolution imaging. And it turns out there's ways of collecting vaso contrast and bold contrast at the same time, two different acquisitions. And it turns out that they're very different sensitivities. And this is not something that we still don't fully understand why, but with blood volume changes, it's much more localized to small vessels really close to the area of activation. So there's more localization, just like with perfusion imaging, than with bold. And here's an example of going to higher and higher resolution. This is 2 millimeters. This is 1 millimeter, 0.75 millimeters. And if you blow that up, you start to see a structure, a change in the structure of the activation. It goes from blobby to these two lines that show layer activation with finger tapping. And it turns out that vaso, I'm not going to go into this, is very sensitive. It kind of jumps out of the sensitivity curve in terms of sensitivity versus specificity. And so that's why we use vaso a lot for looking at layer activation. And that's where I'll leave it with that, because there'll be a whole talk on layer for high resolution or vaso for high resolution imaging, or maybe comparing vaso and bold. A big problem with doing layer fMRI is that you are very sensitive to larger vessel effects. And so bold might give you more sensitivity, but it doesn't give you the specificity where it really matters. So I talked about the whole vein debate in the 90s. That actually didn't matter that much because you're looking at these big blobs anyway. So it never really, but made a difference really. However, when you're going to layer fMRI, it's critical because you have these peel vessels on the top of your layers, and that will completely throw off your interpretation of the signal, of where it's localized in the layer if you rely on bold contrast. And where it is on the layer, cortical layers, really does determine how you interpret the data. Upper and lower layers typically receive feedback from other areas. Middle layers receive input from sensory input, and so on. So the layer activity really makes a difference. You really can't be thrown off by large vessels skewing your signal. It makes much more of a difference at high resolution. So this is just showing you what I just talked about. And I'd like to end with this plot, this diagram. And I'm not going to talk about it at all, but it's just to kind of give you a perspective that that what we're measuring is this right here. And there's many ways we can measure things. We can look at magnitudes, latencies, shapes, the transients, the NMR phase, which I didn't even talk about, and the locations. And you have influences of noise. You have many different pulse sequences that I didn't even really touch upon that much, but I touched on most of these that can modulate what the signal is and how it's related to what you measure. But then you have this hemodynamic response function that smooths the signal, but it also varies depending on how you capture the vasculature with your voxels. And that will skew the results. That will cause differences in what you see on a voxel-wise basis. But there's all this physiology that goes in the signal, and some of it doesn't matter. Like hematocrit kind of matters, but that it certainly matters, but it doesn't vary enough to really throw off your signal. your interpretation. But there's all these different things that can influence the signal. So that's two sides to that. It influences the signal, which could cause you to misinterpret the signal, or could be an opportunity for extracting physiologic information from the signal. So it's a double-edged sword. And then of course, there's many different types of neural activity. I was just talking about activation. Typically, it's local field potentials that people assume, and that would be the oscillations. But we're still trying to probe inhibition and excitation, or transient versus tonic, or we're trying to understand the nature of the spontaneous activity. And then from layer fMRI, we're looking at feed forward versus feedback activity. So there's many different types of neural activity as well that we're trying to extract from this measured signal. So this sort of sizes up the nature of our problem, the nature of the challenge that's still ongoing today. And if you want to, very last slide, if you want to see kind of a nice snapshot into some of the methods development that was done, there's a really nice neuroimage special issue. And I think that there will be a 40 years of fMRI in a few years, but this is a 20 years of fMRI. In 2012, it came out in neuroimage. So that has just a compilation of 100 articles on all the things that I talked about, like how things advanced and more, of course, everything from processing to the physiology and more. So it's a nice reference. It's fun to read. And that's it. All right, thank you.